guests. Hi there. I was just about to tell my good friend Katie the story of Dottie the Carbon Molecule.
I've heard this story before. Carry on.
Right. So, millions of years ago, a carbon molecule named Dottie was floating around the atmosphere in a molecule of carbon dioxide.
Millions of years ago. You were in like high school then, right?
College, actually. As I was saying, she was floating around in the atmosphere when suddenly she got sucked into the stomata of a plant called a tree star.
Ooh! So Dottie got taken in by a producer?
Yep.
Is this my cue to explain photosynthesis?
Yep.
Ugh, you nerd.
There's antimony, arsenic, aluminum, selenium, and hydrogen and oxygen and nitrogen and rhenium, and nickel, neodymium, neptunium, germanium, and iron, americium, ruthenium, uranium, europium, zirconium, lutetium, vanadium, and lanthanum, and osmium, and astatine, and radium, and gold, and protactinium, and indium, and gallium.
Ugh, this is so stupid.
Okay, class, let's begin. Here is the Calvin cycle, the second step of photosynthesis, which occurs in the stroma of a chloroplast, in this case, the chloroplast of a tree star. Here is Dottie. She's incorporated into one of the three CO2 molecules entering carbon fixation. Carbon fixation occurs when the three CO2s are combined with a 5-carbon sugar called RUBP by an enzyme called rubisco. These are combined to make 3-phosphoglycerate. Then 6 ATP are added to phosphorylate this sugar into this sugar, 1,3-biphosphoglycerate. Then to reduce it, 6 NADPHs add electrons to this sugar, and 6 phosphates are taken away. This gets us 6 of the glyceraldehyde 3-phosphates, or G3P. Out of all 6 of those, only 1 will go through the Calvin cycle. 5 will remain in the Calvin cycle, where 3 more ATP are added to regenerate the 5-carbon sugar RUBP for the next cycle. Now, as it happens, the one G3P that left the cycle happened to be the one containing Dottie, our carbon atom. So Dottie, in this little G3P, this G3P was combined with another G3P to make glucose, and then many glucose monomers came together to form starch, a polysaccharide that was then stored in the central vacuole of a mesophyll cell of the tree star. Any questions?
You are such a nerd.
La la la. Then the tree star was picked up by a cute but slightly brain-damaged dinosaur named Littlefoot.
And they called him Littlefoot.
Littlefoot carried the tree star all around Pangea until he got hungry and decided to eat it. He munched on the tree star and digested it.
Well, how does that work?
Ugh!
Beryllium and barium. Isn't that interesting? I hope you're all paying attention. There's going to be a short quiz later on. There's holmium and helium and hafnium and erbium and phosphorus and francium and fluorine and terbium and manganese and mercury.
I can't believe there's more.
So, who wants to learn about how Littlefoot digested the tree star? Okay, let's do this. So, when Littlefoot ate the tree star, it went into his oral cavity. That's his mouth, but we like to give it a fancy name. When he chewed it up, salivary amylase, which is an enzyme, went into his mouth and broke down the starches that were in the tree star. Then it became a bolus, which is just another fancy word for chewed up tree star. He swallowed it, and it went down his esophagus through peristalsis. Then it ended up in his stomach. Now, despite what everyone thinks, Digestion of starch does not actually happen in the stomach. Instead, starches get broken down into acid kind because of enzymes. More enzymes. Everyone loves enzymes. Come on. Don't you love enzymes?
Who doesn't?
I know! So, after the starch is turned into acid kind, it goes down into the small intestine, where pancreatic amylase, which is another enzyme, enzymes, breaks down the starch, which is acid kind, into glucose. And so that glucose is absorbed through the small intestine and sent to muscle cells, where it is incorporated into glycogen. Isn't this so awesome? This is so awesome! Oh, is she sleeping?
Oh my gosh, I had a horrible dream that you were explaining complicated procedures that I don't understand!
But I was.
Ugh. At least you're done.
Not quite.
That she's back in the atmosphere, drops of Jupiter in her head.
So you guys want to know what happened next?
Sure.
So, Littlefoot saw the Great Valley, and he got so excited that he started running towards it. That meant his muscle cells needed to tap into their glycogen reserves for energy. Isn't that so cool?
Pretty cool, yeah.
Yawn! Enzymes hydrolyze the glycogen molecule that Dottie was in, breaking it down into glucose monomers. Those monomers entered the process of cellular respiration. Would you like to do the honors, Katie?
Would I?
Platinum, plutonium, palladium, promethium, potassium, polonium, and tantalum, technetium, titanium, tellurium, and cadmium, and calcium, and chromium, and curium.
Okay, let's begin. So as we know, Dottie, our carbon atom, was part of glucose. Glucose went into this process of glycolysis, which is 10 steps, and it converted the glucose into pyruvate. Pyruvate then went through the pre-Krebs cycle and was converted into acetyl-CoA, and then it entered the Krebs cycle. Through many complex reactions, many things took place, but what's important to us is that carbon dioxide left through this process. So the removal of this carbon dioxide molecule included the removal of Dottie. So Dottie went into the bloodstream of Littlefoot and eventually reached the capillaries enmeshing his alveoli in his lungs. They diffused into his windpipe and he exhaled Dottie.
Great Valley.
Good story. That was only the beginning.
Dun dun dun!
So, Dottie floated around in the atmosphere for a few millennia until she entered the stomata of a birch tree at Katmai National Park in Alaska. There she was fixed and stored as a starch molecule in one of the tree's cells for several years. In 2002, Dottie was moving through the phloem of one of the tree's branches when it fell off during a bad storm.